So protein domain, I think you can think about it as something that was, that's the way I like to think about it. There are many, many more ways to discuss it, but I like to think about it as a successful experiment of evolution that really worked well. So somehow this sequence had the capacity to fold, to retain, and to be recurrent in evolution. So because I think a lot about evolution, I believe that it's a good definition to what is it protein domain. So protein structure, which we didn't discuss explicitly, are really linked to understanding the concept of domain families. And domain families are really linked to the multiple sequence alignment that you just discussed shortly. Now, having a 3D structure is the best way to define a domain and even the domain boundary and so on. However, the problem is that we do not have many of those. Despite the huge increase in the number, this is what is missing in many, many proteins, especially, by the way, specific type of proteins such as membranous protein and so on that are harder to get a high quality structure, okay? So now we are taking the lead to overcome this difficulty. Okay, so again, okay, now it will work. This is a very important slide, very naively stated, but it is about the twilight zone, in which very, naively, one can say that if you have two sequences, we discussed the sequence alignment, that are of a length of, let's say, 100 amino acids, and they have above 40% similarity, the same definition that we talked last week, they are probably related in function, in structure, in protein family domain. Those are safe area. Anything between 20 to 30 or 22, this is called twilight zone, and the effort now that I'll tell you about, which is implemented by Pfam, is really to be able to push the border from the safe zone to the twilight zone. This is the trick. However, what I didn't show you is that most proteins lives here, not here. This is a very narrow, not that many protein lives here. The millions protein that we talked about in UniProt lives here. So moving the border is very important. This is a very informative point. Now, protein classification as a whole can be done in more than one way. I just put the very important resources for structure base, which is Cas and Scope and superfamily. The sequence base, ProtonNet is our own development, SMART and so on, but there are not that many of them. Maybe you'll understand why in a minute. Domain base is Pfam, ProCyte, ProDom, and so on. We'll discuss Pfam, which is really the most, I would say, established and exhaustive. And there are efforts to talk about evolutionary rather than domain-based to see what was conserved and not conserved evolutionary. Okay. So protein family, as I said, those are a group of evolutionary related protein, and the key is that they are coming somehow from common ancestor, and this is what we try to capture, okay? Now, they should have some sequence similarity that is above their randomization or above something, so that we can assign this, trace back the evolution. And it is all based on what we learn quite deeply on sequence alignment methods. Now, just to give you an example before going to the detail, how it looks like. Those are proteins. Here in this very graphical view, what you see are domains, right? This is a protein sequence. Now, this sequence have many, many of this blue domain, right? This is the size of the protein, but you can see quite easily that this specific domain is also incorporated in other proteins with very many different combinations. This is the key. If you understand this view, you understand what's a protein domain family. Domain family is this one, the domain in which it connected the same structure slash a model, statistical model, to explain this blue domain. Now, as I said, usually those are also foldable protein region. So in a very naive way, if I would cut it with a scissor, let them by themselves be in a tube, they will fold nicely. Namely, they do not need any help from the neighbors to be folded. This is a good, also evolutionary-based structural definition that I like. So foldable by itself. Okay. This is another view, just to show you that immediately you can say, if you had to guess what's going on in here, you can see that many of those proteins have this domain, which is called PDZ. Sometimes it's called the zip area, the zip, the zip like a mail zip, because it knows where protein should be anchored. It's a very important one. And you can see it appears in many different combinations. Okay, so Pfam, indeed, it's a protein family. I'm giving you a few highlight on the current Pfam, which is a year old. It has 18,000 libraries, namely entries. Each entry is this blue circle, right? Or this shape that I just showed. Each family has also multiple sequence alignment and a statistical model that we called it HMM or profile HMM, the multiple sequence alignment. What does maybe I'll say a few words about the HMM when I'll show you an example, but I must say that it is really, in my view, the best resource for matching structure, function, and so on. It's really good. Okay. Now, the Pfam is a large collection of protein domain families, so think about it as a library. Okay, you have really library, so that intuitively every new sequence that came from space, you can take it and ask which model fits better this sequence, right? Even that it was not part of this library. That's the ability of Pfam to generalize. Yes.
It relies on multiple sequence elements and the statistical models.
Yeah, yeah. So behind each and every Pfam, out of the 18,000 that I just mentioned, there is a model, which is a statistical model. It's a matrix with statistical analysis of how much I'm, surprised, or how much is. So really, the model is what you are looking for. So whenever you come with a protein, you do not do a multiple sequence alignment. You do a model fitting, and that's important point. And by the way, because of your question, some of those, it's unstable in the sense that if you have another new organism, I don't know, the alligator, and now you refine the model and you see that one of those domain should be split into two because of those new information. So there is this manual updating all the time, and I'll show you an example. But so don't think about it as a one time I did it and that's it, because it's really very sensitive to what's going on in the current data. So the key evolutionary relation, it's determined by a seed. A seed is a small set of multiple sequence alignment that can generalize to the entire set, right? So it is built on a smaller, much smaller set, and we'll see example. The seed alignment form the basis for build a profile, what I called HMM or profile HMM, using the Hammer software. Now, think about it, I'm sure that by now you understand it, but I think it's important to know. When I talked last week, every amino acids or every nucleotide have the same importance. We gave them a penalty and not penalty, but we didn't have a feeling for each and every one of them. Today, class, when Nadav was talking about regular expression, you already noted that some can be much more important than other. This is what is trying to be captured in the HMM. So the HMM really captured the fact that some amino acids are not important to the model. You can just throw them, you know, like by dice. But other you cannot touch. And this is the HMM, and this is why it's so powerful. Now, the profile HMM is queried against a sequence database called a subset that they took from ref proteome. So they didn't take any proteome. They have their own set, which include, by the way, the entire Swiss prot and so on. So it's a big set. And all matches scoring above a curator threshold, that's important. There is will be assigned to this profile HMM, right? So now we understand now a few definition. A family is a domain family, not a protein family. That's important because sometimes it's confusing, okay? Pfam A is the good Pfam. Now, last year, they tried to get rid of Pfam B, but if you go to a previous release, you'll see still Pfam B. B are the ongoing, not yet defined, and sometimes they were destroyed, but Pfam A are the good ones. Pfam are representative to build the model, and HMM is based on the seed. Now, a few more definitions. There is a definition of composition and the definition of architecture. It's very intuitive. The composition is not ordered. So a protein can have domain A 2 times, domain B 3 times, and that's it. And you don't know what's the order. But the architecture is ordered. So you have them from left to right, okay? So you see that it's 2 different definitions. Now, Pfam always look like PF 0, 0, but it's only, I put 2 extra 0, 1, 2, 3, 4, 5, and that's it. So you can, this is a ID that is unchangeable, and it's classified to one of the 6 ways. The 2 big ones are family and domains. Big ones, I say, from the 18,000, let's say, 16,000 or 17,000 belong to them, to this. Family and domain. Family, it's a collection of related protein regions that are big enough and often capture the entire proteins. Domain is a structural unit that I said about this foldability and so on. Repeats are small, short, unstable, that can be multiple copy are presented. I'm not crazy about this definition, but they are also trying to get rid of it, because what's repeat? 6 times is good or 3 times is good. So I don't like this definition, but it's still there. And motifs are important because they are outside of the globular domain. So motif can be a patch in between 2 globular domains, okay? So that's important. Coil is also very important. Again, I don't know how much structure you understand, but coil is really something that has its own self-repetition, and it's a very well-structured and important point. And disordered, despite the fact that you might think that it's less important, it's extremely important. And why is that? I just give you a biological intuition. First of all, disordered is it's not random. To make a sequence being disordered means cannot be folded. You have, or not cannot be folded, but it's not by default folded, to a structure. You cannot just throw amino acids. You have to keep them to be very specific. So the disorder account for about 40% of the proteins in higher organism, and maybe 10% in bacteria, nothing. So there is this disorder expansion in higher eukaryotes, very important, but those are non-globular. So this is the definition. As I already said, this is important. From all the sequence in UniProt KB, 77 do have a Pfam. When I say do have a Pfam, at least one Pfam. However, 53 of all residues fall within Pfam domain. You understand the difference? I'll explain it better. Here I'm asking, if I take million proteins, how many from these million proteins in UniProt KB have any Pfam domain? And the answer is 77% of the proteins. But now I'm asking about coverage. If I take and do a contact, just one huge protein out of this million, and I ask how many patches are covered, it's only 53. So you see the difference. You got it? Okay, good. Pfam B, forget it. Another definition that is important is clan. Clan are Pfam that are related in a way, in a statistical way, so they can be clustered together into something that is not necessarily evolutionary conserved. Sometimes it's converged evolution that are put together. So this is important as well, and I think this is how it looks. I'll give you an example, and just to show you this, I think visually it's very nice to see. This is a real domain of Pfam, the domain family. Now, only the blue one are conserved. So you can say, well, what's going on? I mean, it's probably garbage, but it's not, despite the fact that there are a lot of holes and so on. And this brings us to the last part before the demonstration on the HMM logo. Now, you saw one example of it, but think about it as the following. We have in each and every site here 20 options, right? Now we are going from number 1 to number 25. And immediately, visually, if I have to ask you, it's not, by the way, a great model, but if I have to ask you, what is the most important feature in this sequence, what would you say? You would probably say that, let me say it differently. The higher the letter, it means that information is higher. So it's all been transferred into bits, into information bits. So because you think the baseline model is a statistical model, so you ask yourself, how much is it different from a stochastic event that would just throw those amino acids by random, or not random, but by some prior knowledge on their frequency and so on, okay? So immediately you see that the G and the R and so on, but immediately you see that this is less important. Now, the rule is that the highest, the one on top, is the most informative one. So here you have either tryptophan or tyrosine, almost the same size. This is a little bit more, okay? So immediately you get this feeling, and maybe I'll go to demonstration. Okay, you have, if you have more time, you can really look at the PowerPoint, because I put extra so you'll get good examples of what domain can do, how domains were conserved in evolution and so on. But let's go to the demonstration, and maybe we'll go back if needed. So I'll okay. So we'll go to is it this or that? Never mind. We'll go to Pfam. And you go to the site. Now, as I said, it's a very informative and very good site. And immediately when you see it, you look at the top. You have home, which tells you everything what I told you up to now. You have search, that will go in a minute. You'll have browse, which I like to open now. You have many different ways for FTP, as you already know. A fantastic help and something about. So let's start by the browse. Now, why did I start by the browse? Because if you know nothing, you can have already a very good understanding of what Pfam can provide you. So for example, you can either say something, for example, PDZ that I mentioned, or C2, or any of the domain, or even if you don't know, you just write ATP, and it will give you all the options for ATP. But before I do this, I want to show you that the families are ABC. So if I do, if you have, that it's a helicase, but you don't know which one, you can browse all the helicases. But they do have something very nice, which is the new one. So everything that from the last version to this version, they put it aside so you can really update yourself what is actually going on in the new version, or a new domain, which is kind of nice. They do have the top 20, and they do have also the proteome. If you want the Homo sapiens or specific proteome, you can go from there. So let's just go from whatever, new, okay. Just to give you the feeling. If I'll go to new, I'll get a table. Now, before going to an example, I want you to see this table, because I think this table is super informative. What does it mean? It means, I'll go very fast. Can you see it good enough? Okay. So you see the ID, which is let's say, informative ID. You see the Pfam numbers. You see if it's a domain or repeats, as I mentioned, domain, repeat, family, and so on. But what you see here, and that's very important, is what is the difference between the seed that I explained and the full. And for example, let me show you this example. The seed was only less than 50 protein, but it can generalize to 1,500. On the other hand, there are seeds like 7 that cannot be expanded more than 20, right? So it tells us already something about the, not the percentage, but how much the logo, let's say, the logo capture the entire protein domain family. And you can see that there are really a very big difference between some domains and so on. The other things that we can see is what's the average length of the domain, and immediately you can see that it's mostly below 100. Okay, it's between 60 to 120. And now you see the average. Now the average here, remember the twilight zone that I discussed? You have 30 and so on, but you have also 19, and you have also 60, but you have also 14, and so on. You see a wide range of percentage. And then you see in the last version how much of the coverage. So from the so how much is it covered from the protein, and whether it has a structure or not, and more information. So if we just take one of those, let's take this guy that I have no idea what it is. We get to the page, to the Pfam page. This is the Pfam page that you can get from the search. Now, the reason that I didn't show you the search is because in the search you need to plug in either name or whatever, but they allow now to put a a collection of genes or collection of domains, which is very nice also in the search. But what you see in this family, you have three things: the domain organization that we already discussed, the clan. In this case, they don't have a clan, so you see it kind of dimmed. The alignment that I'll show you, the logo that I'll show you, and few other information. This specific one doesn't have a structure, doesn't have interaction, so it's a fairly poor example. But nevertheless, let's go for it. So we have three architecture. What does it mean? It means always the domain that we are discussing now will be in green, okay? I took a little bit poor, but you can see this. So they say that they have 206 sequences that have this architecture, which is basically a unique one, right? Only this. And they give, if you go you can open it and see all the show all, you can see them all, but let's skip it. And then you have 4 in which they have this and this domain, which is, as you can see, cut, which means that it's not a complete domain. it doesn't fit the entire model, but only part of the model. This is why they do it as if it's broken. I'll show you in a minute. And this is another broken domain. If it's not broken, if it's complete, it will be on this shape of, on this shape. Now, they let you take the entire list of the sequences in a very nice way. They said, okay, do you want only the seed or the full? If you want the seed, you can have it, right? So let's take the view of the seed. Immediately, they'll show you the it should be, okay, let's, okay, somehow, okay, there is this Java business that, do you want, okay, let's see if it works. Okay, let's, go on. So you can see that you can get the HTML, for example, that will work. So you see that the sequence is not that great here, but this is the sequence alignment, right, of this family, this new one that no one knows what it is. So you can see that they have this, and immediately you see that there are a lot of openings, so break and space, but there are some good region of similarity, and the colors really present some kind of code for the coloring, and you can have also anything comes with a very nice way and many different way to download them, and you can see also if you want the full, the seed, the full, or any different proteome according to the similarity level, and the uniprot and so on. So it's a very nice kind of download and alignment. Let's go to the logo. And the logo is the one that I explained very briefly. But what you can see is that the logo, the new type of logo that they have, it's based on skyline. It's called skyline because it's like, the shape of a sky of a city or so on. But immediately you see that they have 3 lines. What does it mean? You understand already what does it mean when I go by sequence by amino acids by amino acids, and you can see that there is here a very substantial, because the bit is close to 4. 4 is almost not the maximum, but it's close to the maximum that you can get. And you can see here arginine and so on, and the rest is really poor. But what they have here nicely embedded in Skyline is that in this region, they have a high probability for opening. And they write you exactly in red that it's a high probability of opening. So this red line say that here there is a space. And this the other lines say how big is the space, how extended is the space. So first you get a code for whether you open it, then some numbers for how likely it is to be open, and what's even the length, the statistical length. So you have a model that is compressed versus the one that I showed you in the alignment. It's compressed, but all the information is captured by 3 lines of the model. Okay, those are the numbers below. Now, I don't want to deal too much on the numbers. You can always see the coloring and understand that this is high probability, low probability. So they give you a very nice hint of how to use the model. And as you can see, this is a rather poor model. Again, it has this nice patch of and another patch and so on. So this is the model of this new one that we talked about. And the last thing that I want to show you is this organism-based story. So for example, this one, immediately you see that it's only in bacteria because it's red. And it's in 2 types of bacteria. If you click here, immediately it will let you download only the stuff that you clicked on. which is beautiful. I mean, you can really go and say, okay, I'm interested in this specific archaea. This specific archaea, no problem, one species, one. So you can mark them, and immediately you can download them directly. So you can either align or generate a FASTA file from the subset that you are on top of them, okay? Now, it's a very visual way, and sometimes just by looking at it, you say, whoa, this is a domain that it's bacteria, archaea, viruses, what's going on? And some domains are very boring, some are very diverse, and so on. So it's a very nice way. To those that do not like too much of this fancy, really fancy, as you can see, very fancy graphics, you can always go to the tree. The tree is the boring way, but it's very informative and very good one, in which everything that I showed you before, it's now in numbers. And again, very beautifully, you can save and take everything from this list. So for example, sometimes you can just click on this and get only saved this subset of sequences. So you can really either hide it or summarize or save or clock or unclock and so on. So maybe I'll stop here. I'll just say without showing you, it's a very, very rich and extremely good resource, really. 3 things that I want to mention about this resource. And one, that it's extremely good for understanding your sequence structure. And I want to add that the multiple sequence alignment that they're doing with the help of curators are used the structure if they do have one. So the structure is always in the seed. That's one point. Second, that they do have a Pfam scan, or in which you can throw your proteins or a set of protein and get the model that fits, including the graphics. So any proteins that you have, use Pfam scan, which is a very good one. The last things that I want to say is that they decide on purpose, which was a very clever decision 20 years ago when they started, not to do overlap between domains. Not because it's not existing, it does exist, but for simplicity and for Python users. So domains are split, separated. They could have cases in which a domain is within the domain, but you'll see it in the graph. And each domain has its own kind of way to show it, but they do not do overlap like the others. The others, when I said the others, is Interpro. Interpro is a combination of 13 different resources, comes from evolution, structure, and regular expression, prosite, and so on. And they combined it all and put a quality control over the outcome. And now, only now, I never liked to use it because it was with a lot of overlap. You couldn't understand what to do. But now they have a very nice addition, which is only for Elixir, and I must compliment them for doing it. Now you can choose out of the 13 which one you want to see the interpro through. So you can say, I want only structural view. So you can click on the 3 or 4 structural resources and see how they agree, and so on. So interpro became back to business and become to be very, very useful as well. Still, you can see interpro through only Pfam. So they talk very nicely to each other. So being in interpro or Pfam will be the same for this on the new implementation of interpro. Questions? Sorry that I rushed, but unless you want me to show you real nice domain, or we'll skip it. So, question. I should just say that the implementation of these 3 lines of statistics for the opening really make the use of the logo very, very fun. Because before it was compressed, they tried to compress it, it was unreadable. And it was a very nice idea. They implemented it in 2014 and it's really took over. And again, you can use your own sequences. I don't know if they have in the web tools, and build your own kind of logo. So it's also an open tool for everybody.